Good morning. I'm gonna do something that's not on the agenda this morning. I'm gonna ask Michelle, a deputy executive director for the Mary program. She's gonna ask a few questions, and whoever wins gets a chance to win a $100 gift card. Are you interested? Say aye. Okay, all right, okay. And that's, I think, was it what's the saying? The early bird gets what? Okay, good. So give yourselves a hand and be ready, okay? All right. Michelle,
Okay.
remember that they're professionals. They know almost everything.
I know, it's going to be
But that's
difficult.
gonna be a hard one.
So, guess we always have to be ready, right? Good morning, everyone. And I think I've met pretty much everybody in here. Who have I met? Would you raise your hand? Yay! So, for those of you that I haven't had the opportunity to meet, I am Michelle Wilson. I proudly and gladly serve as the Executive Deputy Director of MMERI. And I'm happy to be here, and we are always excited about talking about MMERI. So thank you for your time this morning, and thank my boss for his impromptness. So let's go with the question. Let me think. It's got to be hard.
It's got to be hard. Or you can make it two questions and see.
Two questions. Let me see.
Or we could take a vote and split it and say $100 gift card or 50s.
Oh boy, this is hard because you all know everything.
You'd be surprised.
Oh shoot, I can't ask that question.
Ask them what they know about conversations on cannabis.
What was the last conversation on cannabis topic for MMERI?
That's a hard one. I think someone raised their hand.
That's one. That wasn't the last one though. So far, at least you named one. Anyone else? You're close. Anyone else? It starts with marijuana and M. And I'll give you another clue. Dr. Janester Wilson King was our guest. What did you say? Say it again. Yay! One more question?
Yeah, you could do another one.
Okay, let's see. That was a good one. What's the next one?
I think the next one could be.
Just whisper. Don't say it. Just
Okay.
whisper. Aha. So we have several components of MMERI. Our mission is to educate Floridians about the use of medical marijuana, but also about the unlawful use. So that's what makes us unique in the space. We're the only institution in Florida that has that mission. So my question today is, does MMERI conduct research? And if so, what type of research? That's a hard one.
Research, yes. Consumer trends?
No?
The first part, yeah.
Yes. Did everybody hear the first part? Research, yes. Okay. Let me give you a clue. Education starts with a P for the technical term. Starts with a P. I'll explain the technical term.
There's a hand up.
Oh, where's the hand? No, that's a good one though, Dr. Sachdeva. And let's see.
It's another elite term.
It's an elite term for how do you learn?
It's an elite term for education.
Yep, I said education, elite term for education. How do you learn? No. What's another cue? Don't.
let's give it to him and
Okay.
ask another question.
All right, it's pedagogical. Pedagogical, sorry, pedagogical. It's a hard word. And so we're doing research on how best to educate the populations of Florida so that can enhance our ability to communicate and to make sure Floridians understand how best to learn about the use of medical marijuana, but also the unlawful side, of course. We're balanced. Anything else you want to add to that, GP? And then I have to ask another question.
Then we could, I think we could ask them, what are 5 things you know about Mary, and then we could see who answers, who gives the most, and then we go from there. If you can, 5 things, and you win a $100 gift card from one of our partners. I think we should clarify that. The $100 gift cards they don't come from the government, they come from our partners.
Thank you. Okay, I think I saw your hand. What's your name? Tracy. You're affiliated with FAMU? Yes, FAMU. You have a monthly podcast.
Funded by the legislature.
That is correct.
That's correct.
Medical marijuana card.
So you got 1, 2.
What about the medical marijuana card?
Anthony works for you.
Okay,
That's the 2 point.
yeah,
Okay, 3.
that's true.
All right.
She has 3.
And she has,
You have, what else?
how many does she have?
I think 2. Did you have 2, Tracy?
She had 2.
And you
have
3.
She has 3.
Funding comes from the portion of the card state money?
I said that.
We already got that one. Thank you, though.
Okay, Jody's winning.
We're at 3.
Jody and Julie.
Come on. I know it's early. There are some gimmies. The educate.
Yeah, primarily focused on underserved populations.
All Floridians.
Okay.
You were close.
You were close. 2 more. 2 more. Who is this?
Feel free to cheat. Go on the website.
Yeah.
Find something.
3. We're at 3. Quickly. We conduct research. 4. 1 more. One more.
Statewide outreach through coordinated.
Statewide outreach. Good job. You got 2 cards for her? Did she win 2 cards?
Oh, no. She won. Yeah,
Who
she got
won?
1. Jody? Yeah.
Okay,
Yeah.
I got you.
Let's give them a hand. You just might win another one at the end of the presentation, so just listen carefully. My name is GP Mendie. I'm the Assistant Vice President for Research at Florida A&M University, and also the Executive Director of MMERI. Basically, what that means is I just do whatever they tell me. It's quite a privilege to be here, and I really want to thank Gene and his team for always going above and beyond to get us together. So, thank you. Thank you. And when you see Ron, give him a high five, okay? And also look at the next person next to you and give him a high five too, please. Thank you for being here. Thank you. Thank you. Thank you. All right. Yeah, I think Ron is dressed down today. You know, he's not wearing a suit. So, so thank you so much. One of the benefits for the MMERI program when we come here is we get to interact with you guys. And our slogan is educate, learn, talk. We believe that we have a responsibility to educate as mandated by the legislature. We also believe that we have an obligation to learn from the people, Floridians, you guys, as we interact with you and the general public. Nobody knows everything. No one person knows it all. We also believe that we must it is imperative that we keep talking about cannabis. 10 years ago, we couldn't meet like this. Can you imagine? 10 years ago. Look at how far we've come. I want you to give yourselves a hand, really. We've come a long way. And don't forget, we're still in the South, talking about weed. So I want to put it in perspective, you know? And here we are. It doesn't look like we've made a lot of progress, but we have. And your patients and Floridians are benefiting from it. And so, thank you for all you do, really, thank you. Our role is to educate and help Floridians make informed decisions about what's best for them. By law, we're not allowed to take sides. So even if I have an opinion, I'll keep it to myself. Okay? When I'm off the clock, I'm still on the clock. If I don't trust you, I'm not going to express my opinion to you. Because there might be an open mic, you know, just listening. And so it is quite a pleasure to be here. And one of the things that we've been blessed with by the legislature is the ability to be the only entity to educate Floridians about this. We get calls from other states, hey, how are you doing it? What's happening? Come show us how you do it. Because we're the only program in the nation, can you believe it, that's sponsored by a state entity to educate their citizens about marijuana, medical or not. Other states have done it differently, mostly caring about the profit side of it and who's going to get the license and who's not going to get it, what populations are going to get it and what population is not. But educating its people on responsible use was not a priority for them. And I can tell you, I've been talked to people from Colorado, attended conferences in California and Nevada and other states, and Washington, the state of Washington. It is a mess, okay? What state entities, state officials over there would tell you in person, they can't say it on camera so they don't lose their jobs and their pensions. But it is a mess. And here we are in Florida doing an amazing job. It may not feel like that to you, but we are when you compare us to what's happening with other states. So thank you for what you're doing. Thank you for putting your heart into it. Thank you for loving your patients the way you do. And don't forget, as I used to tell doctors and nurses and physicians that used to work for me when I was a COO at a medical company, no matter how much you love your patients, don't forget they can also sue you. So think about the liability, no matter how much you love them when you practice medicine. So we love them. We want to do our best for them. And one of the ways we do that is to educate them. And that's what we do. So please thank you. And Michelle probably has been talking to a lot of you about partnering with us on different endeavors. I want to thank all of you that have partnered with us and have said yes. Barry is on speed dial, you know, and we could call him anytime. And if you want us to add you to that speed dial list, please let us know. And a lot of you have worked with us, and we want to thank you for that. And so it's quite a pleasure to be introducing Dr. Mandip Sachdeva. He's a professor in the College of Pharmacy and Pharmaceutical Sciences at Florida A&M University. I usually tell our researchers, hey, don't forget, in your world, you are a celebrity, okay? There's a world where it's a niche world, and the kind of work you do, and the kind of research you do, and the kind of problems you tackle, you are a hero and a celebrity in that world. And so he belongs to a niche set of group, 2% in the whole world, that are most published and recognized in that field. That's quite a fit. Let's give him a hand as he presents to us today. Dr. Mandip Sachdeva.
All right. Thank you. Thank you, my friend. Good morning. Good morning for coming early, and good morning and good wishes for winning the raffle, those who won it, right? Wish we had more raffles, one for everybody, but hey, that's the future. Okay, so I really thank for the invitation to come here to talk to you guys. This is my first time here, and I thank you, Mary and GP and Michelle, to give me the opportunity. So as you know, for the generous introduction too, thank you for that. So I am a researcher, and I do research in cannabinoids. I do research in drug delivery to deliver cannabinoids more effectively, and I do other things than cannabinoids. However, I realize that we need more evidence-based therapeutics. So in the last, and I've been involved with the consortium, the Marijuana Consortium. I present every year there. My students present there. We just came from there. And I saw that we need more evidence that this works in patients. So with that in mind, you know, I transitioned to the clinical side. That I have to do clinical studies, and I'll talk as I go along that we will be hopefully conducting in 2 or 3 months the phase 1 study with oral cannabinoids for the use of neuropathy, DAP-induced neuropathy. So I got IND and everything else done, just getting the TMH IRB. So with that, background about myself, that I'm a transition guy from research, but I can't go off research. I love research. That's the basis of our future applications for a clinical thing. So I'm gonna start, you know, basically that what is that we are, you heard Mary, we have a mission, right? And you already heard that mission and the vision that we have is that to ensure that all the people in Florida, they, you know, get to know more about the unlawful use of marijuana. So, and I am part of that mission and the vision, which you know, is there, and of course, we had a raffle for this. There are 4 goals, as you all learned: education, community engagement, communication, and of course, pedagogical research. So I'm trying to fit from pedagogical research and to educate and then to get the community going in the process. So, you know, and these are the various activities which have been going on through Mary, and which, you know, we have that at different events, you know, we have made our presence felt. And of course, as I said, that evidence-based research is very, very critical to us, and that's what we are trying to do. Of course, you know, we believe in the entourage effect of the cannabinoids, that there is that a combination is better than one single cannabinoid, minor or major. And that's why the clinical trial I'm doing is comparing a CBD isolate to a full spectrum CBD, because there we have that. Now, I want to go a bit deeper into it. Now, this is a bit of research. It'll be some cell culture and animal data, which is the most driver factor here. So here, that we were very intrigued that the terpenes enhance the permeation of other cannabinoids to the membranes, because they can disturb the membranes and they can do that stuff. Plus, terpenes have a different mechanism than it is found with the cannabinoids. You know, they work on toll-like receptors also, and other such mechanisms are there. So I was very, very keen to look at the combinations of CBD and BC, or no, CBC and BC. Now, CBC is another minor cannabinoid, but getting importance now. CBC and THC have the same chemical formula, same formula, but different groups over there. Number of atoms is totally the same, but different functional groups. THC binds to CB1, whereas CBC binds to CB2. There's no CB1. So because of that, it does not have that high, et cetera, you get with THC, but you get similar benefits with CBC. So CBC was, and I was reading some anecdotal literature that CBC is very interesting and could be a THC possible alternative, possibility to be used. So I thought, okay, let me try. There's a lot of data with CBD everywhere. Let me try CBC and BC and CBC and CBD. So what we did for that is that we basically screened a bunch of terpenes for anti-cancer activity. Here, this data is mostly cancer-related. And there will be some data will be neuropathy-related. So I have 2D models in cell culture. I have 3D model in cell culture. Okay? So here, basically what you see is that I have taken a bunch of cannabinoids, and I chose only one terpene, beta-carotene. It says BC over there. I found from all the terpenes, and there's a lot of data I have, which I'm not showing here, that from all the terpenes, we found out that the beta-carotene is the most stable to be used, and is the most potent, as compared to myrcene or limonene. There's so many terpenes that we tried all of them. And here we are trying different combinations. And you can see here that when you combine CBC and BC, or CBD plus BC, that you see that here we tried with different cell lines. That we had tried a PDX, lung cells, breast cells, and then we tried also lung cells. We found that for different cell lines, this combination of CBC and BC was very effective at very low doses. So that means that BC is helping CBC or CBD to improve its efficacy. So, and the same data we got in 3D culture. I'm not showing it. I'm going to zoom down directly to the animal data. So we said, let's do an experiment in which we took CBC 50 milligram per kg, 1.5, BC 100 milligram. That's the thing. The BC has a higher dose requirement than the cannabinoids, but they are effective at a certain dose. So these were given IP, and then we gave a combination of the same doses. They're not mentioned there, the same doses together. And you can see here, now, we start the therapy, it says day 0, but this is day 14 or 15 because we inject cells into animals, and when the tumors become about 100 millimeter cube, we start therapy. We don't start therapy by just after injecting. We let the tumor grow a bit and then do it. So what we are seeing here is basically this is day 15, and this is like day 30 or something that we are going up to. You can see here that all of the treatments that CBC by itself, BC by itself, so CBC or BC by itself were not that effective as compared to the control. You combine them together, it looks like the BC is helping CBC permeate more, and the process go to the tumor more, and we see that there is a big reduction in tumor growth over here. So, but I said, okay, this is great data, and looks like we have to do more work. So, and to look at what is the mechanism happening over there. The key is mechanism. If you find the mechanism, you can look for other entities to follow the same path and treat your tumor. But you can see that there is a, you can see it's plotted over here on this side that there's a big decrease of the tumor burden as you combine them, okay? Which is great. So then I said, look at some possible mechanism in vitro. We haven't, this is mostly in vitro data, and you can see here that various pathways, the MAP kinase pathways, there is PARP cleavage, apoptosis. This glypican is a very, very key marker in breast cancer. That was highly, and the combination was downregulated. The last, if you see CBC-BC, is showing that there's a lot of suppression of these markers, and they're showing that there's induction of apoptosis. So all these things are happening. We thought that that's good data. But I thought that since the dose is high, the acceptability may not be that much. So I said, let me go to CBC and CBD. Now, all this data I'm showing you is unpublished. So it's like a garden conference where we are supposed to only present unpublished work, not work done 5 years ago or whatever. So this all is in data. So then I said, let me go to CBC, CBD. So when you combine two drugs together, you have to know what's the right ratio. It's something called isobolographic analysis, where you vary one, keep one constant, and vice versa, and see what's going on. And then if you see that the graph or the line graph is moving inwards, that shows more potency is happening compared to the each. So you see over here that here we combine CBC and BC, and so these numbers are the IC50 of each of the drugs combined. Here, in this case, we use half the dose of CBC and varied CBD. See that what's the CBD IC50 now? You see how much drop is there from, to 0.8. That means that there's several folds, improvement with half the dose of CBC. So that means if you combine them in a certain ratio, you can have synergism. That's what you want. Not additive effect, but synergism. So, this we showed in different cell lines. We have a lot of cell lines which are resistant to doxorubicin, resistant to Taxol or paclitaxel. So, and then we have breast cancer cells which are letrozole resistant, MCF7 cell line. So we have all these because the key is to show how does your product work with resistant cell lines. And what we found out, now I haven't shown you a lot of data with the wild type, but in the wild type cell lines, these cannabinoids work okay. But when we go to a resistant cell line, something unique happens, which we are finding out, possibly epigenetics. A lot of histone modification are occurring, which are not seen in the wild type cells. So, and we are working, I have some data maybe, but we are working on the ATAC sequencing and other analysis to find out the new targets in epigenetics, which is a very underserved area. But anyways, moving forward, here we looking at various cell lines. The MCGA-B2 is a PDX, a patient-derived cell line. So in all these cell lines, you see that this green line over here is moved way inward, showing that there's significant synergism when to combine these two together, which is great. And then we also showed that there is reduce in the cell migration, because that's one thing that you want to prevent the migration. And in the combination of CBC, CBD, we see that significant decrease in the cell migration, So, and then we want to make our point with that is this happening only in one cell line or in multiple cell lines? So here I'm showing you data, again, mechanistic, this is Western blot data, that in this MDAMB 468 doxorubicin resistant cells. These cells have a very high IC50 for doxorubicin. And we give cannabinoids, they become responsive. So something really great is happening. If this translates to clinic, that'll be like a great thing to help those patients. So anyway, so here also, looking, I'm trying to show that when we combine cannabinoids, how various pathways are better activated. Here again, I'm looking at the mTOR pathway. I'm looking at NF-kappa B, SOT2. NF-kappa B is a key marker for inflammation. And then we are looking at other markers for apoptosis over here. So basically, and also the mTOR pathway and the AKT pathway, you see that in the combination, you see a significant reduction in those activities, showing that there's a tremendous synergism happening if you see that all the Western blot. This is just a, beta-actin, which is the housekeeping gene which we have to have. So, and again, looking at another cell line, which is MCGB2 DOX-resistant cell line, which is a PDX, a patient-derived cell line. We show that it's the same thing happening here, NF-kappa B, SAW2, and same way for the ERK pathway was also, highly, impacted. And then looking at the AKT and looking at the other markers for apoptosis. So basically the point I'm making is that this is not a one cell line phenomenon. This combination of CBD and CBC is a very potent combination. So this is all in vitro. Now we said, let's go into animal studies and see what happens there. So we found out that the ratio of 2:1 is very essential to see this effect. So if you have 2 times CBD and 1 time CBC, that's a great combination. And that's what we kept over here. If you see here that we took a 20 milligram per kg of CBD and 10 milligram per kg of CBC. We did them individually, and then we also combined them. And you see here that in the combination, which is this line graph over here, you see that the significant suppression of the tumor burden compared to the control, which is it keeps growing. Or other, by themselves, they are okay. Combine them, 2:1 ratio, something great happens. And then we also looked at the various mechanisms, like looking at vimentin, which is a metastatic marker, which is highly downregulated, as you see here in the combination, because these are 4 lanes. The last lane is for the combination. And same thing for the other markers like caspase or the cleat-PARP. They all are hallmark of apoptosis. There's increased apoptosis because that's what leads to cell death. And then the mTOR or phospho-AKT, that we see that there's a lot of pathways you saw in vitro are translating in vivo, as we see here. And then we tried to use one more model, which is a 231, which is another cell line. And again, you see here that the CBC-CBD combination, again, controls tumor. These are very, very resistant cells. They grow like wildfire, that become huge. So to control them below 1,000 millimeter cube is a great achievement. And then again, various pathways which were implicated, like vimentin again, and survivin, apoptotic marker, PARP, and the kappa B inflammatory, mTOR. So all these apoptosis pathways were highly activated in the process. So we have heard a lot about the role of CYP enzymes and the CYP inhibition yesterday. And there were speakers from TrueLeaf who explained very well. There are CYP, 2C9, 2C19, and other CYP enzymes which are inhibited that can lead to improved PK of other drugs combined with it. So both CBC and CBD are CYP inhibitors. They have some common CYP enzymes, CYP2C9, CYP2C90, which they both inhibit. So there'll be a competitive inhibition going on when both of them are put together in some ratio. So we want to understand its kinetics, that what's happening in the PK, and can we validate by using some validation model, like we have this GastroPlus, a simulation model, which can do in silico modeling and see that are your data validated. So here, this is again a lot of data in one slide that we did experiment with rats and check for the kinetics, that how is the kinetics of this combination as compared to by themselves. And as you see here, that look at the Cmax over here. Now, we gave these formulations orally and also by IP. We gave both the routes. But I'm going to talk about orally just to keep the time going. So this is the oral data, right? And you can see here that when you combine them together, CBC and BC, given orally, at the ratio of 2:1, there's significant increase of the kinetics. For example, if you see here, the Cmax, the highest concentration which we get in circulation, the Cmax, at a certain time point, which is all given here, that's the key thing, that when you combine CBC with CBD, there is about 1.5-fold increase in the Cmax of the CBC. And for the CBD, there is a increase in twofold. There's a twofold increase over here, as you see here, for the Cmax for CBD. So 1.5-fold for CBC and 2-fold for CBD. That means that there's more drug in circulation. And why this is happening is mostly because of the comparative inhibition for the same CYP enzyme. So there's more CBD, and in the kinetics way it works, that will inhibit more. Then the CBC goes up. CBC is an equally potent compound. So in the process, CBD also goes up in the process. And then as you see these benefits, there's a combination floating in circulation, and that can do a better job and sustain it for a longer time. That's why you see the tumor growth was controlled for a longer time. Now, you could improve this by making a formulation. It could be a lipid base or a micellar base or a, liposome base or exosome base. You can use different formulation strategies to make it better, right? You can make a self-emulsifying system orally. You can do a lot of different delivery approaches, which we are doing for different projects. So anyway, so that's the point I'm making that. And then when we use in silico analysis by using GastroPlus, doing simulations, by using, they have a model they give you based on the human physiology, and then you can put the numbers in there, what you're getting, and then you see that if your model is validated or not. And then we found out here that for the data, the solid line which is showing here is for the validation. The data we got in the other color of the line we have compared to what was we were getting by validation by this software, that the data was highly validated, and this confirmed that there's more amount of, say, CBD in the jejunum, in duodenum and jejunum, the main sites of absorption orally. And the same way, there's more amount of CBC when you combine them, as you see here, that in those parts of the gut. So that means that we could validate this model, and and you could use more validations using other softwares, but we use GastroPlus, and we're going to use SimCip also and see how we can show that this is really happening. So anyways, do this very quickly, that since I'm working also with pain, use of cannabinoids in pain, and I have developed two models. One is chemotherapy-induced neuropathy by using paclitaxel given to animals, and then it develops neuropathy if you give it for 7 days in a certain dose. And then we look for the behavior parameters in animals. We have all the equipment for that. And then you can, do your treatment and look at the change in the behavior parameters, and then you can do some mechanistic study with the tissues you get. That's one model I have. Second is the diabetes-induced neuropathy model. And our clinical trial will be on the diabetes-induced neuropathy patients. So I have both models. I'm going to just show you only very quickly 1 or 2 slides. The point I'm making is that since I'm a delivery guy, so I get drawn into delivery a lot. I don't know if you guys have heard about exosomes. The exosomes are those communicating bodies in the human being. For cell-to-cell communication, you need something that how cells talk to each other, and that's done through exosomes. Exosomes are everywhere. They are in the milk, they are in your human body, and they are, in the fruits. You can get exosome from all sources. That means that the exosome has the package information of your DNA, RNA, all this thing, and it's transporting. Exosomes are there in tumor cells. Exosomes are there in the good cells, which are growing. So maybe you have heard about it, that there's a exosomal kit on the market to look for your PSA levels from your urine, because your urine is full of exosomes, because they are cell communicating, and then you have to take out something out of your body. So exosomes are everywhere. Exosomes are used as a biomarkers for disease. There's a big research going on, a lot of companies spinning off on exosomes. So I've used exosomes from stem cells, mesenchymal stem cells. I have got exosomes from natural killer cells, and I have got exosomes from retinal organoids. Now people are talking of organoids, everything, but you can get exosome from organoid. They are better representing of that organ. So, and I have also got exosome from milk. Believe it or not, you can get exosome from milk. There's a big industry on that and make into various cosmetic products, and you can combine that with cannabinoids. The people have formed companies on that. So anyways, in this, I'm going to talk about very quickly of mesenchymal stem cells derived exosomes, which I did. And we have to have a bioreactor, scale it up, and we have to characterize it. It's a steps process, a technology, right? So, and then we made formulation of cannabinoids in exosomes that we made a formulation. And we use that for this model of chemotherapy-induced neuropathy. So we gave that formulation IP to the animal so that it will go everywhere in the body. You could have given it also IV, but we gave it IP for the sake of convenience, and then looked at the behavior parameters. Look at that if you give a stimulus to animal, how fast it withdraws its paw. Because if the animal has neuropathy, it will have a slow time in withdrawing because, hey, I'm hurting, I cannot withdraw. If the animal is healthy, it will withdraw it very quickly. So we give a score to it, and there are various hot and cold and different methods, different techniques to do it. So that's my basis of it. So here what we did was that we use a CBD by itself. There's no combination here, but CBD in exosomes. CBD in exosomes. And then we gave it to animals. We gave CBD alone, exosome alone, and then we looked at the behavior parameters, which are shown over here. But this one shows a bit better the whole. So here it shows the paw withdrawal latency, right? In hot and cold stimuli. Right? People behave differently to hot and cold stimuli. So it's good to see that. So anyways, so if you look at the control here, we give it a score because that you get from the equipment. A score is given. So here we see that it's a full score of 10 or more or in that bracket. But as you apply this paclitaxel, this the next row, the score drops because the animal has neuropathy, and it won't withdraw that fast. So, and that's what the score we are seeing here. And then when we use the EVs, we have made EVs as the exosome. We made the formulation of exosomes. When you make that, there is some, very little improvement that shows that exosomes themselves are not doing a lot, and then little bit. And then we combine this with CBD at 5 milligram per kg dose. We see that some change, and when we take this paclitaxel, treated animals with exosome by themselves plus CBD, right? Plain exosome plus CBD, that we see some. But here you see that when you put this into the exosomes, the CBD, as a formulation, sustained delivery, you see that the score goes back to what the score was to the normal animal, very, very close score. That means that by making a delivery system, you can reduce neuropathy in a model system, or maybe it can translate to the clinical side that a delivery system may help, especially exosomes, which themselves have some potency in reducing neuropathy. MSC exosomes are very, very popular. A lot of applications. You see, if you look on the Google, a lot of formulation being sold with MSC exosome for wound healing, for different applications, right? But they have a big role to play in this also. But as a combination in a delivery system with exosomes, it really works very well. So and then we looked at the various mechanisms. So we think that it is going through the AMPK pathway through SIRT1 and NERV1 and NERV2, upregulation, and to the mitochondrial function and biogenesis. That this was our, and this work is still ongoing in the lab. So here, let's go to more that all this, information we have, how we can use this for pedagogical research, how we can help the community and the pharmaceutical, the industry working in cannabis, and also the clinical side. How can this all be brought up to speed into these areas? So basically, we already talked about that. What is the goal? Is to address the challenges, which we have about that. What we need to know more about medical marijuana, and to look at this unlawful use being going on these days, and how that's impacting the people in Florida and beyond. So and of course, our goal is to have always an evidence-based education. So basically, how do you want to do this? And our goal is to use a cutting-edge, educational strategies, and using some interdisciplinary methodologies also, so that we can, look at or understand more that what is the things happening for the unlawful use of medical marijuana. This research which we are now starting at FAMU in this direction, or we have been doing some work in the past, but this is another direction we are going, is that to enhance public understanding of the risk and the benefits and pharmacological differences between marijuana or THC taken, on a regular basis or on an unlawful basis, how that does really impact. So we have, different goals and different objectives over here, and the goals here basically is to strengthen or to understand more about the health risk, especially in the cardiovascular area. There's a big concern that when we are taking cardiovascular drugs along with cannabinoids, or cannabinoids themselves may have some myocardial effects, which is of a great concern and going on these days. And also, it's important for long-term testing of the cannabinoids, sitting in a container, sitting on a shelf, sitting in a bottle, that what's the long-term stability issues with that. And also that how we can develop in the process the evidence-based curricula, so that we can better look at the populations which are vulnerable for these effects. So, I had gone to a USP conference like last month, and USP and FDA are very interested now to come with guidelines. And the way USP works, that they every year they come with a new monograph for different things. There's a monograph for cannabinoids. And I met the folks there who designed the monograph for cannabinoids, and there are amendments made every year. So, and they came out with the monograph last year, which is online. You can go and look at it. And then this year, they came out with some new changes for the water content, for the limits of how much you have to have the cannabinoids. And they leave these changes for public debate and feedback till they implement it. So we have till July of this year to give our feedback. So for the new guidelines that come out from USP, which they will import on the FDA, and so they work all together. So, so please do have a look at those new changes they are trying to implement for the new monograph to come out, because once it comes out, that becomes the Bible to follow for everybody here. So please give your feedback to what, which they have, so that we can. But anyways, so this is from the monograph they have so far, but there are some changes coming up that, and this is what they are saying, that treat cannabis as a single highly variable species, which is basically we have been talking about it, and since THC is one of the commonly used, combination with CBD, that the ratio that you have to establish a formal order of the express ratio, And also, the definition of THC dominant, CBD dominant, and intermediate chemotypes. That's another, highlight over there. And the container must be labeled with the name and the amount of any cannabinoid present in the amount of 10 milligram per kg or more. And use the labeled amount of all cannabinoids measured milligram per gram, where the contents are no less than 80% and no more than 120%. That's it's going to change. 80-20 is going to change to 91-10, right? So we have to keep those changes coming up, that they will have us more flexibility, but it's changing as we go along. And then there's also a big concern on the microbial contamination limits. What are the contaminants and what is the microbial contamination? Big concern that we need to have. And the products must be labeled if they have gone any microbial remediation. If something was done in the facility for something, that has to be fully disclosed. So these are so these are the changes, plus more coming, which you should have a look and see. Okay, so what are our research goals? That we have different, specific aims over here, and that we want to do some surveys, and that's why I want to talk to some of you to talk about how to improve our surveys we are doing, because we are trying to get more data from the public who come to dispensaries, who come to various cannabis shops or otherwise, that why they are doing it, how do they feel, get more data so that we can help the community more. And then also to assess, which is the second name, is to physical characteristics, the stability part of the formulations, and also looking at the contaminants and the risk involved with these formulations. So to do a systematic understanding of all that. And then the third aim is that education, reach out, is to help more community engagement, to give more literacy to the public, who's our main endpoints, that the public should be informed. So of course, we have different, techniques how we're going to do that. Of course, in our lab, for example, in my lab, we have a lot of facilities to analyze things. I have a HPLC, I have a LCMS, and also for gummies, if you pick up gummies from 10 different suppliers, you will see that each of them has a different texture, different feeling how to bite and whatnot. Well, can't we have a uniform standard for that, which should be followed? So we have, so in the lab, we have a texture analyzer. Okay, thank you. I've got 5 minutes to go, okay? And miles to cover, so maybe part 2, right? Make sure you have more gift cards for the part 2, okay? Okay, so in our lab, I make gummies in my lab, I have 3D printed gummies, and I have made gummies in molds, right? So, and we published a paper also with 3D printed gummies. I'm not going to talk about that, but we have a texture analyzer which can tell the texture, the hardness, that how the gummy is, and we can give a data, and then we can say, hey, from these 10 manufacturers, this gummy is the ideal or good one, where this is too hard or too soft or whatever. So there are things you can do which can help the consumer. So that we're gonna do some of that work in the lab. It's pedagogical, that to see that how different gummies have different, behaviors. And then also, we are also concerned about that the stability studies that, at different temperature, at accelerated, sometimes you are traveling in a warm, you say you go to Morocco, it's pretty hard, you are on vacation, you have your CBD gummies with you. Well, how will they behave in that warm weather and it gets warmer, or if you go to any other country which is more warmer and you are outside a lot. So, and also looking at contaminants, that how do we have a role to play? It could be pedagogical, that looking at various COAs from various products and see that what are the limits of contaminants they have, and are they following the guidelines or not? So these are the various, of course, outcomes, that more patient safety, more informed public, and of course, there are long-term public health benefits that we're going to empower the healthcare professionals and the public with the evidence-based information. We're going to be very, very scientific-based and evidence-based, and then give the information out. So this is the whole summary of what we're going to do. How we're going to, work with some science, some pedagogical, a lot of evidence-based, and then inform the public accordingly as to what needs to be done. Thank you. And I still have a couple minutes to spare. Thank you. Thank you.
Let's give him a hand again, please. Thank you. Before I talk about any gift cards, questions? We got 4 minutes. No questions? Please, this session was recorded, so feel free to go back, listen, and look at it again. It's a lot of information that was presented. And it's our job to synthesize that and make it such that the average, not professionals like you, but the average Floridian understands. Please, we have a question back there.
Hi, you just said you were working with the edibles. Have you looked at terpene delivery, like terpenes in the edibles? Like, are they actually there or absorbed, I guess? I've always wondered edible absorption of terpenes in humans.
Correct, and that's a great question. I did not go into the direction of terpenes because they are volatile, most of them, right? They may be in the edibles, in a very small amount. The issue comes up that can you detect them by any analysis in the process? So I thought that terpenes is a good idea, and they have a good future, but they need a higher dose to be effective in combinations. So I did not pursue that direction, but I went back to the minor cannabinoids like CBC. I'm working with CBG also, and CBG has amazing data with pancreatic cancer, which I cannot talk about here today, but we got a fascinating data. We compared CBD and CBG was much better. But to your question, no, we have not looked at the edibles, but if somebody is interested, we can work with them.
Jody here? Sorry. Jody?
Appreciate it, Norbert. First of all, thank you for the work that you're doing in stability testing. I was just astounded when this program began to start, and none of our labs were ready to do stability testing. How do I know what the shelf life is? So I really appreciate that you guys are delving into that. Are you primarily using CBD and minor cannabinoids because of the federal prohibition on THC? And would FAMU status allow you to get through some of those roadblocks to use THC in your studies? Or was that a professional choice or kind of a dictated choice?
Okay. Necessity is the mother of invention, right? So I wanted to work with THC, but we don't have a license at FAMU to that I can have beyond 0.3%. So I said, hey, THC is being used, which is great, but there should be other minor cannabinoids which may act like THC. And so I looked at THCV for a while, and I published a paper with that with THCV, but THCV is expensive, and it's very gooey mass, it's hard to work with, easily get oxidized, whatever. So I have to use my scientific brain. A lot of times you say, this is good, but you gotta see what is the end point? Can this be a product on the shelf? So then I looked at CBC, and I said, oh, that looks interesting. And I went deeper, and that's what I showed you, that you go deeper in science, you get new answers that lead to new applications. And with that, since you brought up a sensitive topic, with CBC and CBD, I went to FDA for a clinical trial that I want to do this combination in neuropathy because I got data, very good data in animals, amazing data. And in cancer also, but cancer is a more difficult trial. So, and they said, well, there's no data on CBC in the literature that much. Tell us how, but they combine them together, what's their PK, right? And I said, well, I have no data for that. So I had to drop that idea for the clinical trial and we have to again renovate and say, okay, I'll do CBD and full spectrum CBD and see that they're minor there, but it's full spectrum and you are used to it. I went back in with that strategy and gave all my 150-page document, and after back and forth, this year I got the IND approved. So you have to re-strategize yourself. So every battle lost is a second battle to win for if you keep thinking. So now I showed you some data with CBD, CBC, which shows that I have the data, right? And we are using all these in silico modeling, which FDA loves, right? So, and I'm gonna go back after I finish this trial, I'll do in one more, like a model, like maybe in a dog or in some higher animal, because they need 2 different species. And then I'm gonna go back, because this idea is not done yet. So, to answer your question finally, that we don't have a license. So if we had that, I could have done more TSC work. Thank you.
Thank you. Ladies and gentlemen, we are out of time, and I apologize that I'll probably talk to Gene about how we can do the next gift card. Don't forget, how do you explain